Ok, et du coup, il faut que je commence de partager mon écran. Clément, you'll be kicked out from the room at some point for the deliberation, so I encourage you to try and follow the defense online on YouTube on the MIND-INRIA YouTube channel.
Okay, sure. Thank you. Sorry about that. I didn't know it was
Yeah,
on the
no worries.
jury thing. Okay.
No worries. Thank you. I think we're good to start.
Great. So hello, everybody. Welcome here today for the defense of Alexis Thual for the title of Doctor in Neurosciences and Applied Mathematics. You will have 45 minutes to present your work, and then you will have questions from the jury members. 4 of them are online. We will start in the following order. We'll start with your 2 rapporteurs, Emma Robinson and then Ruffin van Roelen. Then we'll hear from Wim van Duffel, then Gabrielle Perrier, and then we'll follow with jury members in the room. So Claudia Lunghi, myself, and then we'll have comments from your PhD directors, Bertrand Thirion and Stanislas Deun. Please.
Thank you. Esteemed members of the jury, dear members of the audience, friends in here, it's my pleasure to welcome you to my PhD defense today, where I'll be presenting the work I've been doing for the past few years, and in which I have worked on developing methods to compare cortical surfaces across participants, but also across species. First, we'll start with a bit of background on research that has been happening in the past 3 decades, and that has been trying to map the functional organization of the brain, both in humans, but also in non-human primates. And we'll see how differences in the organization of the cortex are both an impediment and an opportunity to study the brain. And then I'll switch to the contributions that I've tried to bring to the field. So first, we'll be exclusively working with MRI images, which leverage different phenomena from nuclear magnetic resonance. And mostly you can get 2 types of images, the one you can see on the left of this slide, which are called anatomical images, and they give you a sense of how the brain is organized. And on the right, you can see what we call functional images, which are mostly a time series of what's going on in the brain. So mostly what we look at are movements of the blood in cortical tissues, and from that, we try and derive information about what the brain is doing. As you can see on the right of this image, it's not possible with a naked eye to understand what's going on, especially in the right image. So we need to heavily pre-process this image to get anything interesting from them. I won't go into the details of all the pre-processing that needs to be done, but this figure is taken from the fMRI prep package documentation. And so you can infer from it that there are a lot of steps that need to be computed, and one of them is going to be of particular interest in our case, and is highlighted in green, and it's called anatomical co-registration. We'll come back to it later. Moreover, when you want to deal with functional images, most of the time, you'll have to get your hands on statistical models to extract the relevant information from the images collected. So the most classical approach is to try and model the observed and captured images Y as a response to a specific kind of stimuli. The underlying phenomenon is illustrated with what you see in the top right-hand corner in this slide, namely that when an area in the brain is particularly activated by a sort of stimulus, you will see a kick in blood flow a few seconds later in order to bring some oxygen to this part of the cortex. And so if we know, and that's the figure below, if we know the stimuli that the person has seen at a given moment in time, we can try and deduce what part of their brain responds specifically to this sort of stimulus. So what you derive in the end is this matrix of beta coefficients. And in order to gain even more signal, we actually compute linear combinations of these beta coefficients. These combinations are called contrast maps, and this concept is important, and we'll be seeing a lot of contrast maps throughout this presentation. Mostly the idea is that, and this is what you see at the top of this slide, here are brain responses in the same person when they are either reading complex sentences or reading strings made of words that are made up of consonants, so like they don't have a meaning. Obviously, we're more interested in parts of the brain that respond specifically to sentences, so we can combine these two images together and get the one that you see at the bottom. Interestingly, the activations that we could see in the occipital lobe, so the visual part of the brain, fade out, and we mostly get activations in areas 44, 45, which are notoriously known for parsing language. So actually, this endeavor has been going on for 3 decades now, almost, and people have famously used fMRI to try and get a map of the brain. Starting actually before this century, people were using fMRI to show the existence of places in the brain that specialize in the processing of faces or places or even word reading. And so obviously there's been a lot of work going on about this. Like, can you map the whole brain? Are areas in the brain specialized in one kind of stimulus? Can you exhibit parts of the brain that would maybe react to a wide series of a wider range of categories of stimuli? So all these questions remained open for a while and are part of the endeavor of mapping the brain. Obviously, and there's a distinction that I want to make at this stage. A lot of the work that started 3 decades ago mostly relied on classification. So there were specific classes of objects that we were trying to see in the brain, like faces, places, etc. And I think the whole setup somewhat changed when people tried to regress latent representations, so more like continuous information from these stimuli instead of classifying them. In this specific example, Alexander Huth and colleagues showed that they could map the reactions of the brain to specific semantics of language. So they could isolate parts of the brain that would react mostly to violence-related word or places-related word, etc. An interesting problem also. So mostly what I've been showing to you here is what is called encoding. So we try and explain the brain in the sense that we try to explain the dynamics of the brain as responses to specific stimuli. You can see this problem the other way around. So if I give you a picture of the brain, can you deduce what the person is doing or seeing or viewing? This endeavor is called decoding. And the seminal work of James Haxby in 2001 paved the way towards trying to classify what a person is seeing. So in this particular example, we see images of brain activity when a person is seeing a face or a place. And from this maps of brain activity, we would try and reconstruct and guess what the person is doing. Again, major effort has been put to try and map the whole brain as a whole. So we can cite the work of Arthur Mensch and colleagues who trained decoders on the whole brain. And so given a map of the brain, they would try and deduce what the person is doing, but from a general perspective, what task they're performing at the moment. And again, I'd like to go back to the distinction previously made in the previous slide between classification and regression. What we're seeing at the moment is the advent of regression methods for decoding. So people are not just trying to guess the category of objects that you're seeing at the moment. They try and maybe reconstruct the whole image that you would be seeing while being scanned. We can cite the work of Schoen and colleagues, but also Ostelik and Van Roolen. What you see on the left is the work of Schoen and colleagues, where they try and predict low-level features of what a participant would be seeing. Low level meaning, are they seeing a red object, where the red object is located in the image, etc. And more recently, we've seen major progress being made in decoding high-level features. High level being meaning, is there a cat in the picture, or is there movement, or these sort of information, and not only where is the action happening. The thing is, with all that I've presented here, it's a bit hard to draw conclusions at the population level, because we all differ, all our brains are different, and so it's a bit hard to use these tools in a way that's completely generic. Let me give you an example. So if we go back to the contrast map that I've showed you before, which is the map on the left, if we were to do the exact same experiment in a different participant, you would get a map maybe like the one you see on the right. So these are two different individuals, and they're doing the exact same thing. And we can notice two things. First, their anatomy is different. The shape of the brain is not the same. And although they're hearing the same sentences exactly, and we're doing the same statistical processing, we see that the areas of their brain that react to these stimuli are not located in the exact same place and might not do the exact same things. They might not be as activated from one experiment to another, etc. So once we know this, the question of understanding what's going on at the level of, at the population level remains open. Like, what do we do with these images? Classically, people have resorted to using anatomical co-registration, which is what I mentioned in the pre-processing phase. So instead of dealing with individual anatomies, they would make the assumption that everyone can be projected onto a common anatomy, such that then we can perform statistical processing of these images. But let's pause here for a second. So let's say we can precisely locate the fusiform face area, so the area that reacts to faces in participant A. And let's say that we can locate the parahippocampal place area in participant B. How would we know how these areas are located relative to one another? We wouldn't know, because we've collected this data in two different people. So in order to tackle this problem, people have been trying to collect more and more data in a limited number of participants. And that is the endeavor of the Individual Brain Targeting Project, for instance, that has scanned 12 human participants for more than 50 hours each, resulting in a collection of more than 400 contrast maps per participant. What I'm showing you here are screenshots from a web application that I've developed that is called Brain Cockpit, and that offers you to actually deep dive into this data. And so what the application does is you can click on a particular point of someone's brain and see how this particular point in subject 5, can confidently be linked to specific type of stimuli. So this chart right here that describes the functional activity of this voxel is what we call the functional signature or functional fingerprint. It's rich information about what this part of their brain is doing. Obviously, what's interesting is to do this between people. So what we want is to take another subject, go exactly at the same spot in their brain, and see if the functional signature is the same. So this is a rather simple but also naive way of looking at things. However, it has proved very useful. I think I was mostly interested in the seminal work of Dante Mantini and colleagues in 2013, where they've done exactly this. So they've tried to collect rich functional fingerprints, both in humans and monkeys, by having them watch movies. And then they've drawn comparisons between voxels of the monkey cortex and voxels of the human cortex, so as to be able to compare them. Obviously, more advanced techniques have been used as well, and then this point-wise comparison. Obviously, we need to cite the work of James Haxby and colleagues who have developed a general framework that a lot of people have been following for the past few years. Mostly the idea is that if two people do the same experiment, we want to find some sort of transformation of their individual signal that projects all of this information into a common space. Mostly they were using rotations in the functional space. And other people have followed this endeavor with different kinds of optimization models. So we can think of shared response models, but also optimal transport being used to align between brains. However, and we can cite the work of Thomas Bazet and colleagues who have benchmarked all of these methods. It's not clear at this stage what method works best. Also, one cannot but speak about the seminal work of Emma Robinson and colleagues, who have introduced the multimodal surface matching method that is used, again, to align functionally brains instead of simply relying on anatomy. And mostly their method consists in computing continuous deformation field of meshes so as to maximize functional similarity between individuals. Interestingly, this method has been used in interspecies setups. So this functional alignment method has been used, for example, to compare the cortical organization of human and monkey subjects. And in this particular example, they relied on resting state data and tried to highlight parts of the brain in the two species, which could be confidently mapped to functional homologs in the other species. All right, so that's where we start. I've presented mostly two big categories of functional alignment methods: MSM, which is a continuous transformation of the brain and doesn't need parcellations, and hyperalignment-like techniques, in which current solvers for optimal transport would fall, that are not continuous methods, but need somewhat rich priors about the anatomy of the two brains that should be aligned. What we want is to create a new method that would allow non-diffeomorphic transformations between brains, and that does not need prior anatomical data between the compared brains. So with this, I'll start presenting the contributions that we try and bring to the field. I'll present a method that we developed, which hopefully alleviates the previous problems that I mentioned, and we'll see how we can use it to try and get more insights into what we can decode from brain data. So let's go back to intuitively what we want to do. Let's say we have two participants, a source and a target participant, and we've collected a large collection of contrast maps for them. So maybe we ask them to move their right hand, and that's what the contrast map you would see here. Or maybe we ask them to listen to sentences, that the contrast map you would see at the bottom. And we do that for a lot of tasks. Another way to look at this data is to look at it from the vertex perspective. So that's what we've seen in Brain Cockpit. So we can describe this particular vertex right here as its response to a wide series of stimuli. And what we want to compute is what we call an optimal transport, a transformation P, that would map together vertices in the two brains. So in this particular example, it's quite obvious what we want to do. We want to map I with K and J with L. So that's the first ingredient of the optimization problem we're trying to solve. But then let's imagine that there's a voxel in the temporal lobe of the target subject that has a functional signature that's very similar to that of K. Do we have any way of making sure that I will not be mapped to M? And this is the second ingredient of the optimization problem that we're trying to solve. Namely, the Gromov-Wasserstein constraint in this optimization problem will try and preserve the general anatomical organization of the two brains that are being mapped together. More precisely, what it does is it makes sure that if 2 points, 2 vertices, were far away from an anatomical standpoint in the source brain, they should be mapped to points that are also far away in the target brain. And this way, you can do slight changes locally, but the general anatomy of the brain should be preserved. Lastly, we add what is called an unbalancing constraint in optimal transport. And the intuition behind this is that maybe some functional areas are bigger in one brain than the other. Or maybe they don't exist at all. Like for instance, when if we compare humans with macaques, it's very likely that some areas exist only in the human brain and maybe not in the monkey brain. So we want to be able to either shrink or expand functional areas from one brain to the other. Lastly, we add hyperparameters to each of these contributions so as to be able to weight their relative importance. This problem is actually very close to a problem that was very studied extensively by Thibaut Séjourné and colleagues, and for which they find a very elegant way of approximating solutions. So mostly what they said is that this problem is hard to solve because it's non-convex. And mostly coefficients that you see in elements that you see in pink are interactions between coefficients of P, which makes the problem hard to track and hard to solve. So instead, they say, let's not just find one optimal transport, but either two. And we'll replace these interactions between coefficients of P, by interactions between coefficients of P and Q. And what they say is, obviously, these two problems are equivalent if you ask that P and Q are equal. And they actually look for approximation of a lower bound. So they only ask that the sum of coefficients of P and Q should be equal. Interestingly, if you fix one of the two plans, so let's say Q is fixed, solving the lower bound for the other plan, let's say P, is equivalent to solving an optimal transport problem that is very well known. And namely, it's the unbalanced Wasserstein problem, for which solvers already exist online, implementations you can just download, and they would work. So as a summary, you can see the algorithm on the right. Mostly the intuition is we fix P, optimize for Q, fix Q, optimize for P, and repeat. And hopefully this gives us good approximations to the problem we want to solve. At this stage, I want to show you what these matrices are. Like, what is an optimal transport plan? So most intuitively, again, this is in Brain Carpet. We can explore these optimal transport plans here. And what I've done is I've clicked on a human cortex that was mapped to a macaque cortex. And what you see represented on the right here is one line of the optimal transport plan. And what you see is that it could be interpreted as a probability density of the vertices in the monkey that should be mapped to this vertex in the human participant. An interesting property of these plans is that if we click on a voxel that's just nearby, so here, you see that the density map follows along and does not go, just anywhere in the brain. Is that mission solved? So we don't know yet. Qualitatively, what I've just showed you here is the way to look at the plans, but we'd also like to have a quantitative way of evaluating their relevance. In order to do so, what we've done is we've computed a transport plan using 300 maps from the IBC dataset that I presented to you before. And we have evaluated their capacity to predict unseen contrast maps in the target participant. So what we do is, let's say we train our optimal transport plan using experiments where the person was listening to text or like moving their hands, etc. And at test time, we scan the person while they listen to math, for instance, or like completely different tasks. And what we do is we observe their brain. And from this, we can use our transport map to predict what we should see in the target brain if they were to do this math experiment. And luckily, we also collected this data, so we can, compare our prediction to what actually happened in the lab. The baseline we want to compare ourselves to is the case where we don't use functional alignment, but we only rely on anatomical co-registration. So we would only compute the correlation between brains that have been aligned using anatomy. We add one more experiment. We want to see if functional alignment can be used in combination to anatomical co-registration. So what we do is we project all contrast maps from above onto a common anatomical template, and we do the exact same procedure, compute an optimal transport plan, and do predictions. Interestingly, we see that for every pair of individuals of the IBC dataset, the correlation between their brain, once they've been functionally aligned, is significantly higher compared to what the baseline would offer in both cases. So when we rely on anatomical co-registration at the bottom or not at the top. One of the main impediments of these methods, though, is that the number of hyperparameters that we need to tune is actually quite high. And so what I'm showing you here is a cross-validation table where we have varied all possible values of alpha, rho, and epsilon. And I won't go too much into the details. Mostly the idea is that if you put reasonable values for these hyperparameters, you'll get some gain. However, the maximal gain that you can get in terms of correlation is only observable if you tune these hyperparameters correctly. There are many other variables of interest than correlation gain. I won't go into the details here, but the idea that non-trivial values are needed is of crucial importance here. All right, so at this stage, what is interesting is that it would be really hard to do the same experiment with monkeys. So it's really hard to actually derive a protocol in which both human participants and monkey participants would engage. And so if we want to compute alignments between two species, we probably need a different task, a different evaluation. We've resorted to using decoding for a wide variety of reasons, but also this one. So what is decoding? Mostly decoding nowadays consists in doing the following. A participant, human or non-human, is put in the MRI machine while they take part into an experiment. In our case, it's going to be movie watching. So they watch a movie. And what we're trying to do is to predict what they're seeing from their brain data. So the way we do this is we take the movie, have it go through an AI, which is called an image encoder. The image encoder gives us a hopefully useful representation of the information contained in the movie. And then we train an algorithm that is given brain data and that tries to output latent representations of the movie. More concretely, the data that we're using looks like this. So we use data from the Nguyen and colleagues data set that was collected in 3 human participants who have watched short movie clips for more than 4 hours. These movie clips are about 20 seconds long. You can see an accelerated version of them below. And so what we do is we have the data that you see at the top of this slide, and we try and regress latent representations, such as the one that are represented at the bottom. Maybe one key ingredient here that I'd like to precise is that we use rather simple brain decoding models, namely linear models, in this case, read regressions. And in contrast to other decoding efforts that are being pursued at the moment, we strive to decode ongoing information, so on the fly. And to do so, we only minimally pre-process our brain data. So mostly what you see at the center of this slide are time series collected in 5 different points in a human brain in 2 different sessions. And mostly the only 2 steps that we follow here are we detrend all cosine shifts from these trends, and eventually standardize this data such that it looks like what you see on the right. Again, because of the delay in the hemodynamic response in the brain, an image that's seen at time t will only elicit a response that we can see in the brain later on in time. So we'll actually use brain data that was collected later to decode this particular image. How much later is called the lag, and we usually use a lag of 4 seconds. And how many brain volumes is called the window size, and we usually set a window size of 2 brain volumes. So once we have trained a decoder, we need to evaluate it. First, we do this qualitatively by having it play the following game. So when the decoder makes a prediction, we give it 100 images. Only one of them is the true image that the person was seeing. And the decoder has to try and guess which one of these 100 images is the good one. So, it's the model could say, I think this image is the right one, then probably this one, this one, this one. So the model just ranks all possibilities. What we see here in this qualitative example is in the top row, you can see the image that the person was seeing while they were in the scanner. And in the bottom row, you can see the images that were retrieved by the model. So the first line is what the model thinks, what the decoder thinks the person was seeing, et cetera, et cetera, et cetera. In some cases, we get the right image, in the first few tries. But in most cases, we get something that's not the right image, but that is semantically consistent with what the person is seeing. I'll let you appreciate the fact that, for instance, in this column, the person is seeing a soldier, running in a field, and the retrieved images are images of people running by the sea. When they see pictures of animals, we retrieve pictures of animals. Underwater pictures, we retrieve underwater pictures. And this gives us a natural way to try and quantitatively evaluate these methods, these decoders. Namely, for a given prediction y hat, and the ground truth latent representation associated with it y, we rank the similarity of the ground truth image with regard to y hat, with regard to that of every other image of the retrieval set. Namely, it's just we count the number of images in the retrieval set that are better than the ground truth. I've showed you at the bottom of this slide 2 distributions of the rank of ground truth images when we train a random decoder and a linear decoder. What you can see is that when the decoder is random, the rank of the ground truth is just evenly distributed across all possible ranks. But when the decoder is actually learning something, the ground truth image oftentimes pops in the first retrieved images by the decoder. We sum up these distributions by just using the median rank, so with just one number, and the median rank being this bar that you see right here. What we observe is that, so we've tested many different image encoders. I'll come back to that later. But what we see is that it's possible to train decoders in every participant of the dataset that achieve pretty decent decoding accuracy on this task. What's interesting at this stage is that once we have a latent representation, there is a very rich number of applications that we can imagine from there. One of them being putting these predicted latents into a generative model and try and reconstruct an image of what the person should be seeing. So bear with me for a second. At this stage, we've used versatile diffusion, which is an open generative model accessible online. What you see at the top of this slide is what the person is actually seeing, and below are reconstructions with our decoder on the 3 subjects of the one dataset. What you see below were attempts from previous research papers. Again, these images are not, they're not perfect, but they usually get the semantics of what's going on. So here we have this, yogi, latexy, indoorsy vibe of what's going on. We do better with portraits of people or people running, etc. But you get that this is a noisy process, and this is where we are thus far. We've extensively studied the impact of the quantity of data on these results. So if you recall, the median rank is the only number we're interested in that I've presented before, and the lower, the better. And what we see is that with more data, the median rank gets closer to 0, which is good. What is interesting is that had we had more data, the results would be even better. We're not hitting a plateau, so collecting more data would probably yield better decoders. What we've studied as well is we've tried to put ourselves in a one trial decoding setup. That is to say, we evaluate the performance when at test time, the person is seeing the image just once. What people do usually is they report more, more optimistic metrics because they have been testing the participant many times at test phase, and they average these images together so as to get better signal-to-noise ratio. Right, so now is the time to try and bring all this together. So it's notoriously known that these decoders work well. You can train them and test them in one participant and they work well. However, if you test them in a different participant than the one that was used during training, they fail completely. Not completely, but like the performance drops quite significantly. So the idea was to use Fugue, our alignment method, to try if we can bring these decoders to participants that they've never seen before. And mostly it just consists in piping all of these models together. What we see is, so what I've represented in gray here is the performance when we train and test these decoders in the same participant. Just below is the median rank, so the performance, when we use the anatomical co-registration. So you see that the performance drops. And the line just below is when we align test participants to training participants using our Fugue method. And you see that we recover a lot of the performance that was initially present in these decoders. We've also tried more complex setups. So we've also tried training these decoders not only on one participant, but on a group of them. And these lines right here represent if we train these decoders on only anatomically co-registered individuals or functionally aligned individuals. Again, we observe a slight gain compared to what we would do with individually trained decoders. Most interestingly, this gives us a way to transfer decoders to people who have only been in the scanner for a few minutes. So I'll let you observe here that if we only collect 32 minutes of data in a participant, this dot in gray is the performance we would achieve if we were to train a decoder on them, on their data, on their 32 minutes of data only. But we can reach a much better performance if we instead align this participant to someone for whom we have a lot of data accessible. In high quantities in a limited number of participants, and to see how it generalizes to participants more numerous, but who have been less scanned. I'll go quickly on these slides, but mostly the idea is that most of what I have presented here is valid, whatever image encoder you use. So I won't go into the details, but we've replicated these experiments in different setups. So I'll take a pause here just to let you think about what you'd like to do next. And obviously, with what I've presented before, the idea is to try and bring this whole pipeline to interspecies comparison. So the idea is very similar. We're going to train brain decoders in human participants. then we will compute alignments between human and monkey cortices, and pipe all this together so as to see if a brain decoder that was trained in human participants can decode activity going on in the macaque brain. Then we'll do something slightly different from before. So the latents that we've predicted here will be used as inputs to a text generative model. There are many reasons for why we want to use these generative models, but mostly what we will end up with are short sentences describing what the person is seeing, whether it be a human or a monkey. The dataset that we rely on is the following. So for the human data part, it consists of 8 participants from the IBC dataset. For the non-human primate data, it consists in 2 macaques that were scanned in Wim van der Vel's lab. Both species will be looking at a Disney documentary entitled Monkey Kingdom, which is mostly a 1-hour-long movie featuring images of macaques interacting, fighting, eating, etc. This movie will be divided in 5 segments, and human participants will only watch it once, but macaques will see it 3 times so that we get better signal-to-noise ratio. The same human participants will also be have also been scanned while watching short movie clips. So again, it's a very similar setup to what we've seen before. And we use this data. We've seen that the amount of data used to train the decoders is a crucial point. So we try to leverage this to train the best decoders in humans. So first, we can see that training and testing a decoder in human participants in this setup works. So we get pretty much much better than chance decoding performance in human participants, even though we have less training data and we are in a single test data setup. What you see at the bottom are 3 examples that are taken from a retrieval task. So what you see on the left is what the person is actually seeing in the scanner. Then you see a caption that is reconstructed from the ground truth latent representations of these images. And what you see on the right are retrieved images from the retrieval set and generated captions. I think this one is a particularly good example as to what we're trying to do. So the generated caption is a man is trying to pull a stunt on a red car. So you see that it's not grammatically completely correct, etc. And there's no car in the actual image, but we get a lot of the interesting bits of the information. Lastly, what we do is we train a decoder for each human participant. This is what you see on the y-axis of this graph. We train these decoders using the movie clips and the first 2 segments of Monkey Kingdom. Then we use the first 2 segments of Monkey Kingdom to align human brains with monkey brains. And finally, we try and decode a completely unseen part of the movie, namely the 4th segment of the movie. What you see here is the median rank of our decoding distributions, and the last 2 columns are the results for the monkey subjects. What you see is that, again, we achieve better than chance performance in these participants, and that the performance is somewhat comparable to that of human beings. We also run a control experiment where we shuffle our predictions and see that the median rank falls back to chance level, indicating that we're not just predicting large properties of the dataset, but that we're actually predicting things that correlate with what the person is actually seeing at acquisition time. Again, this is a very qualitative example, but I wanted to show you what captions look like when trying to reconstruct from monkey data. And I particularly like this example. So the generated caption for this image right here is a close-up of a face with a picture of a woman in the background. And if you had seen Monkey Kingdom, you would know that the main character is this monkey right here, and it's a female. And I don't know, I think it's interesting that, maybe these models would come up with properties of the stimulus that maybe we wouldn't integrate in our protocol in the first place. So at this stage, I hope that I've convinced you that Fugue is a method that can successfully leverage functional and anatomical data to align data between human participants, but that it can also be used in more challenging setups, such as the decoding of the regression of latent representations of perception, and that these generalization to non-human participants, but also to human participants, but also non-human primates, was interesting to you. Obviously, there are many things I would do differently, and which I think I should frame as future research directions. First, it's really hard to tune the hyperparameters of Fugue. And so I think there's a very useful venue as when it comes to finding a systematic way to weight the relative importance of all components of Fugue. Then, an interesting property of Fugue is that it's a distance, actually. And as a distance, it makes it possible to compute barycenters between brains, which I think is a pretty interesting idea in the sense that it would help maybe building what we think are prototypical brains, both from an anatomical and functional standpoint. Lastly, the Gromov-Westerstein constraint that we've seen in Fugue only has cortical distances, geodesics on the cortex. But there are many interesting sets of data that I would like to use with this constraint, mostly functional connectivity matrices or white matter tracks connections between areas of the brain. Lastly, when it comes to decoding, I hope to have convinced you also, but well, my personal belief is that future research directions that involve the development of new methods are obviously welcome, but the workhorse of this research is actually data. And we'll need more data and more individual data that we have at the moment if we want to pursue in this direction. And lastly, I think maybe one of the most open questions thus far is on the side of MRI sequences, which are at the moment not optimized for decoding purposes. And I think tweaking this in a way that maybe would increase temporal resolution could be an interesting venue. This work from the past 3 years led to publications in journals, but also conferences. And I could attend many conferences in which I participated in the form of posters, etc. I was also lucky to be part of bigger open source projects such as NILearn. And I think this was a very pleasant but also important endeavor in this PhD work. And with that, I'd like to thank my mentors, my contributors, obviously, friends met along the way, and you all in the audience for your attention. Thank you.
Thank you very much, Alexis, for this presentation. I would like now to give the floor to Dr. Emma Robinson from King's College for around 20 minutes.
Okay, thank you. Congratulations, that was a really great talk. I really enjoyed reading your thesis. So I have a number of questions. So the first one I was just noting down at the beginning was I was just interested, you said later on in the decoding that you did minimal pre-processing. Was that both for the decoding dataset and the IBC? What pre-processing was done over? So you said you detrended, but I assume, like, how did you map the fMRI to the volume, and did you not look at any sort of denoising techniques?
So I'll rephrase your question just so that so as to try and make sure that everyone in the audience can hear it. But if I understand correctly, the question is what preprocessing did we do in the decoding phase? And at which stage of the preprocessing did we stop? Maybe of the classical preprocessing did we stop?
Yes, so both for the decoding dataset and for the IBC dataset.
So I think I'll start with the I'll start with the IBC dataset. The IBC dataset mostly goes through the entire pipeline. So we have all the classical preprocessing pipeline, preprocess both anatomical and functional images. And then the data that we used to align between brains is a combination of both conditions and contrast maps. So we do not only use contrast maps, we also have maps of beta coefficients that were simply normalized by the covariance of the beta matrix. But this setup is only valid in the first part of this presentation. So this is the first setup in which we tested Fugue. But in the decoding phase, we actually no longer rely on any contrast whatsoever. We directly use the bold signal collected in human participants. So in this case, we have a TR of 2 seconds, and we make absolutely no assumption as to what the stimulus should be. There is still some pre-processing. I mean, it's not raw data. Obviously, it was reconstructed from case-based sampling, and then there is a slice timing correction. There is also a projection back to an anatomical template, in this case, MNI. And then mostly we used surfaces throughout this work, mostly for simplicity, because it made for a very intuitive and nice way of visualizing our data, of subsampling big meshes, etc. But I think none of what we do has to happen on surfaces, and we would happily go back to volumes had we had the time to do this.
Because the reason I'm asking is that how much there must be still physiological noise in the data, right?
Sorry, can you say again? I'm sorry, that's the sound
Sorry,
of a bit.
I'm trying to speak up. There must still be physiological noise in the data, right? Motion,
Yes.
Respiration. How much is that impacting your mappings?
So we haven't measured this. We haven't tried computing our mappings with and without head motion estimation, for instance, or these sort of things. The IBC data is preprocessed in a way that tries to regress out head motion. It's the same for the one data set, for instance, and it's the same for the monkey data that we've used. So, this is actually something that was done during the preprocessing phase and not something we do with a general linear model where we try to regress this out of the signal. So obviously, yes, you're absolutely right. This is a frontal impediment to what we're trying to do, and these bits of the signal are still present in the time series that we process with our decoders, at least on the monkey side.
Yeah. Because with less without the diffeomorphic constraints, like there is a chance that you're sort of mapping to pockets of noise. I guess the follow-up question to that one was, I believe from your feet, so you tried aligning with different subsets of contrast with that and then validating in others. How much variability in your mappings do you get if you use different subsets of contrast? And how do you ensure that you're aligning with enough contrast to give you good coverage of the entire brain?
So one of the things can you see the slide here?
Yeah.
One of the things we've tried just after submitting the chapter, the paper on Fugue, is to try with different types of training data. And so what you see at the top of this slide is the kind of correlation gain that we can hope for if we use all of IBC. And then we've tried different subsets of this, but also we've tried using movie clips, etc. It's obviously a combinatorial problem, so we could run an enormous grid of setups, and this is I think this is a very interesting research project. You know, maybe trying to come up with the minimal protocol that needs to be reproduced from one lab to another, so as to produce the best functional alignment, while you know, being not maybe acquisition heavy on the MRI side. But this is still an open question. We see, however, that with less contrast maps in this case, the gain is less important. And so, again, what we've tested at this stage is to compare the, and this is part of your question, is to compare the similarity between computed plans. We actually do this with an optimal transport metric. It's not something that I've thoroughly tracked in these experiments, but we would see, we would compute anatomical, functional alignments that are actually pretty close by whatever the training date is. What's happening, the difference between these two lines right here, mostly happens in areas where we gained signal with the new contrast maps, and it wouldn't really disturb the contrast maps already aligned with the Archie protocol, for instance.
Yeah, I guess a visualization of this, not that it's easy, would be quite good, like seeing, I don't know, how much variability there is in the, I don't know, the displacements or the variance of the maps, if you had a consistent test set, maybe. On slide 35, I think there was a clarification, but you were saying that it looks it's I think it was when you were these ones were at the 35 and 36, like the output is like a probability distribution function, but why is that the case, that you get a distribution?
So you're absolutely right. It's not exactly a probability distribution in the sense that it doesn't sum to 1, but you foster it to try and sum to close to 1. It's just that if you renormalize a line of P, the relative importance of all coefficients in the line will remain the same, and it's actually an important piece of information. So what we see here is that some voxels in the monkey brain, you know, ended up with a coefficient that's much higher than the rest of the line, and that's enough regardless of the the norm of the whole line. So you're right. It's not strictly speaking a probability density, but I think it makes for a nice interpretation to see it this way.
Okay, so when you're mapping the data from one to another, does the value of that, the value that's going to voxel 1, 2, 6, 4, the other way around, it spreads out?
Yes, it's very much. Sorry, say again.
The data corresponding to that point sort of gets spread out.
Yes, and in particular, so I don't have slides for this right here, but mostly there's this hyperparameter here, epsilon, that you need to tweak carefully when you use this method. So essentially what would be happening is with different values of epsilon, the spread, like the variance of this probability distribution, would be different.
Okay.
So with smaller or higher values of epsilon, you would end up with larger, groups of red in here, or smaller groups. And so I think this is the main impediment thus far, because tweaking the data is, somewhat relevant, interesting, etc. Whereas, tweaking this value of epsilon is merely a computer sciencey problem and not something that's relevant from the neuroscience part. But you're right that it would impact the images that we see right here a lot.
Okay, that's cool. Oh, so there was just a point, I think I raised this in my, like when you're doing the decoding, so I think it's slide 49, how do you sample the negative samples? Do you just do it randomly? Because you get the returned examples 1 to 5, they're all the same. So I assume it's like, or you know, in that second column, is it just that entire video is just watching someone talk, so it's over a long period of time? Because
Absolutely, yes, that's it. So the retrieval set comprises all ground truth images of the test space. So it's, I don't know, like I think a few dozens of minutes of data. And there's one particular clip where we see this this person talking. And so every 2 seconds we'd get, you know, a movie frame that's very similar to the ones we already have in the data set. Maybe you can see a slight change between retrieval 4 and retrieval 5. But yes, there are redundancies in the retrieval set. And I think that's one way we could probably easily, you know, improve these metrics by making sure that the retrieval set is free from these repetitions, maybe.
yeah, maybe go. I mean, we were trying to do something similar recently, and we sort of set the retrieval so that we weren't sampling within a certain amount of time of the ground truth, because we knew that the frames weren't going to change that much. So well, I think that's fair, because otherwise you're sort of making it look harder for yourself.
Yes, and I think it's still an open question, because we would like to rely on retrieval sets that are not taken from the set of stimuli. We would like to rely on Lion or, you know, enormous collections of images from the internet, but it's very likely that the distribution of these images is different from the one that we learn during training. And so I think this would be doable with more fMRI data, where we actually sample, where we actually show images to participants whose distribution correctly maps that of the test set. But at this stage, with just a few hours of data and just a few hours of movie clips, I think it's very unlikely that we can build a retrieval set that has all the good properties that we could hope for.
No, yeah, it's difficult. Is that what you meant by optimizing the MRI sequences for decoding then?
So I think one of the things I would try right away is unless we work with datasets like the natural scenes datasets, for instance. So for context, the natural scenes dataset is an fMRI dataset that was collected in 4 participants who have been scanned for more than 40 hours, seeing images of the ImageNet dataset. So we have literally thousands of samples of these people watching images. And I think using fMRI with a repetition time of 2 seconds in this setup may make sense. But if you watch a movie, you know, maybe you'd be interested in having not as good a signal to noise ratio, but a higher temporal resolution. And I think the results, the decoding performance we've seen using MEG or EEG recently on massive data sets, you know, is a hint that temporal resolution versus signal to noise ratio is not to the advantage of what we do with fMRI at the moment. So what I'd do is I'd take the same fMRI sequences that we use at the moment. I'd just use an acquisition time that would be much lower and a repetition and echo time that would be much lower as well. Just a repetition time, sorry.
Yeah. Sorry. I was just thinking so this might be going off on a tangent a bit, but like, so you were comparing to like the MEG work by Jean King, I suppose. And like, do you think that temporal resolution isn't everything, right? fMRI gives you something about the spatial autocorrelation of the data. You know, I guess you weren't using per se in your model, but do you think that no, there is an advantage to using this fMRI in terms of spatial resolution?
Yes, definitely. And maybe not only spatial resolution, but I think obviously the advantage with regard to EEG is that with fMRI, we can collect data in parts of the brain that are much deeper than just the outer part of the cortex. And I think it's very likely that this brings interesting bits in the signal when it comes to decoding. quite frankly, and I'm not, I just don't know what part of the cortex contributes to decoding what we decode. So I've tried inverting these decoders. One of the main reasons why we chose to work with rich regressions here was with the hope that these matrices could be inverted and somewhat act as encoders of the brain, and that once this model is trained, if it's inverted, then we can try and rediscover the fusiform face area and then ask more complex questions about the brain. The thing is, it doesn't seem to work like this. So without constraints on these models to actually rely on the entire signal of a functional area, I think they're bound to maybe relying on just a couple of voxels to try and decode specific categories of perceived visual images. And so this is where we stopped. But I think my intuition is that an ingredient is missing on the decoding side to try and maybe leverage all of the features that you were mentioning.
Yeah. So then slide 59, where you're comparing the multi-subject trained to the single subject trained. When you compare the anatomical alignment and the Phu, are they both being tested in unseen subjects? And
sorry, can you say again? Are they
the two bits at the bottom, the green ones, are these both being tested in unseen subjects?
They're tested in the same subjects. You're right. So here we train one model using all three participants of the data set. So subjects 1, 2, 3, and we measure the decoding performance within in subject 1, and then subject 2, and then subject 3. So this is just one model. The line below, again, is just one decoder tested individually on each individual. So these two all green lines are within subject setups.
And you see less improvement here between anatomical to functionally aligned.
Yes. I think this is, so again, at this stage, personally, I think it's hard to know if the model is learning anything that's common across individuals, or if it's using some coefficients to fit the first subject, then some coefficients to fit the second subject, etc. So I don't know if it's really one model or three models in one, because we have too many coefficients. And so what I've tried in the past few weeks is to replace these linear models with deep learning models that have a huge bottleneck. So, or like a small bottleneck, depending on how you see it, but like deep learning models that force a big contraction of the dimension of data. And what I could see is that when using deep learning models as decoders, very shallow deep learning models, and very lightweight, I could bring some more gain very significantly. Maybe in a way, maybe because it forces the model to leverage signal in a common fashion across subjects.
No, that makes sense. And then I guess a lot of the experiments you've done have been with, especially the decoding, you have data sets where you've got very few subjects, but lots of long recordings. Have you considered or tried training and testing in the HCP data where you've got less contrast and shorter movies, but more subjects?
This is ongoing work, but it's not my work. Pierre-Louis in the lab is using Fugue at the moment to compute barycenters of individuals of the HCP dataset. And in this way, it makes it possible to try and train a classifier on all individuals at the same time, because they've been aligned together in a common space. But it's not something that I've tried personally, and it's not published work at the moment.
What do you mean by barycenters? Actually, I didn't follow that. Do you mean by barycenters, do you mean sort of the centers of clusters or?
Yes, so by barycenters, I mean something like this. So if we could find a distribution of functional signal on a barycenter, on a barycentric brain, we would like to make sure that this object, so this map of the brain, their functional signatures in each voxel. And this is what the same map would look like using using a fugue barycenter. I've received a message that my internet connection is unstable. Did you
Yeah,
hear
I
us?
think every now and then you sort of drop out, but
Oh yeah, every 30 minutes.
I thought it was me, but no, it's good because I'd forgotten. I remember reading about this in the thesis, so that's okay. That's pretty much it. I had one quick simple question on the monkeys, or the macaques, which is why is the data much more noisy? Why did you do 3 runs?
Why is the data more noisy? So I think I wrote that here. Open. So it's more noisy in the macaques. Sorry, I don't know what slide in particular you're referring to, but more generally, this data is noisier because human participants were not fixating. So there is eye gaze during Monkey Kingdom compared to what we see usually with natural scenes dataset or datasets of this kind. And again, it's very well known, although these monkeys have been scanned using Mayan and not Bold, the signal to noise ratio is less advantageous in monkeys in this particular dataset, but also in other movie watching dataset. So, and it's always a bit hard because then you may want them to rewatch the segments of the movie many more times, but then it's a bit hard, you know, what's going on in their head when they see the movie for like the 6th time. So we decided to go with a rather small number of runs.
Okay.
Thank
And
you.
just average it after anatomical alignment.
Yes.
Cool.
Thank you very much, Dr. Robinson, for the questions. We will now move to our next rapporteur, Ruffin van Roffel from
Thank you.
the CERCO in Toulouse. And again, you have around 20 minutes for the questions.
Yes, thank you. Can you hear me well?
Yes, very well.
Okay. So I want to start by congratulating you on the manuscript and on the presentation. I really enjoyed reading the manuscript and I learned a lot. I actually did not know about your FUG, I suppose that's how you pronounce it, your FUG method. So I learned about it. I was very happy, and it's very clear, very well presented. And also I appreciated your presentation in which you selected the important subsets of the manuscript and of your work instead of trying to cram everything into one presentation. So this made it very clear. And so here are now my questions, and I think I'm going to try to go from more specific, more detailed questions or more technical questions to more generic ones. And the first question that I had, which unfortunately does not refer to anything you presented, it was about the mapping between humans and monkeys. But I think the question also applies to mapping between different humans. I was surprised when I looked at the results because I found them counterintuitive. So you have a figure, it's the figure 5.4, but it doesn't matter. You have a figure where we see different source subjects, human source subjects, and different monkey target subjects. And it felt to me, yeah, you just passed it. Yeah, this one. And it felt to me that mostly what we see here is that the results seem to reflect idiosyncrasies of the source more than of the target, in the sense that when I compare macaque 1, the projection onto macaque 1 from the 3 different humans, they all tend to look the same. And oh, sorry, no, the other way around. Macaque ones from the different humans do tend to look different. But I see the same thing in macaque 2 and see the same thing in macaque 3, as if really what matters in the projection, in the mapping, is the source rather than the target. And I would have thought that it should be the other way around, that when I'm projecting something onto a particular macaque brain, which is different from another one, then probably I want to capture something about this particular macaque brain. And then I realized there's no reason why there should be an asymmetry, so maybe it should be a bit of both. But then there is an asymmetry here, and I was wondering if you could comment on this.
Yes. So I think there are many interesting points here. I'll just start by showing that the problem in the way we framed it is completely symmetric. So if we were to switch the T indices with the S indices, it should not change any of the quantities that we're evaluating here. This hopefully is also a property of the solver, but it's not something that I've thoroughly checked. And so this problem right here is not convex. And we derive a plan P, then a plan Q, etc. iteratively. I've tried different initializations to try and see if we would converge to the same solutions, and that's what I get in general. But I've never checked that swapping the source and target subjects would actually yield the same plans. So this would be something we'd like to do. However, if I have to go back to this particular example, I think it's also important maybe to, so I don't want to try and oversell this figure. Actually, one of the reasons why we switched to decoding instead of doing what we're doing here is because this data set maybe was not the best to do what we wanted to do. And so I wouldn't take these figures too seriously from a functional perspective here, because they are leveraging a data set that, even though it's been collected in different participants and uses several modality, features functional signatures that are a lot less rich than what we have with movie watching. I think, so mostly what we wanted to do here is to show that we could actually scale optimal transport solvers to meshes with a very high number of vertices on matrices that wouldn't fit on a GPU. And actually, I think the main conclusion here would be that, things are just too clean in the prefrontal cortex, for instance, etc. So these plans obviously are relying mostly on anatomy more than features. So what would be coherent to do at this stage is to probably rerun parts of these results, but they're not published. So this is why we didn't go back to doing this. But now that I have a lot more insight on how Fugue works, what I would do is I would compute these plans again and drastically decrease the importance that's given to the anatomical part in the Fugue loss. At this stage in my PhD, it was not obvious that this was the right thing to do, but now truly this is what I would do. But maybe also to be very clear what's going on here, I've built intuition over the past few years as to what is a good plan. And I'm very sorry to say that usually a good plan, a good quality transportation plan is usually one that does not suit neuroanatomists. So if the image is too clean, it's a bit suspicious. And I'd like to go and show you maybe a picture of what a quantitatively good plan actually looks like. So these are, this is mostly the same figure than the one you've seen before. So we plot the atlas on a flat representation of the human brain, and then we see how this is, how this colorization is mapped onto the monkey brain. I've tested different hyperparameters here. I'll just move that around so that people can watch the numbers. But it's a bit disappointing maybe to neuroanatomists that the best plan in the crowd is this one right here. So, it shuffles things around a lot. And I think computationally, this has been one of the reasons why people using hyperalignment, for instance, or optimal transport have relied on parcellations. It was one way of, preventing voxels from jumping around, etc. And from one way of making sure that their transformations would be, more conservative. If you release these constraints and solely rely on data, this is the kind of results that you get. So I don't think this is I don't think we're doomed to having plans that look like this. I just think that the response is not going to be found on the methodological side of things. I think we need to scan the same individuals more so as to enrich the description that we can make of each and every part of their brain.
Thank you. One important question that I had when I read your manuscript and that you actually answered in the presentation, and I actually did not mention it in my review. So I was very happy that you anticipated my question. My main question was why, when you decode, you only use the latents for retrievals, or actually sometimes for captioning, and never for image reconstruction, even though we know that we have fairly good models nowadays for image reconstruction. And I've done that a lot myself. So I know how to evaluate these results. And in the intervening time, you did go and play a bit with the image reconstruction. I was very happy to see these results. I find them fairly convincing. I just had one question about this. You say you used versatile diffusion, which make it sound a little bit like what we did also with Furcano-Zelik in the brain diffuser paper. Yeah. Maybe with one difference, in the sense that we initialized our image reconstruction with a low level guess from a VDVA autoencoder, and that tended to make it didn't change the semantics of the reconstructed image, but it allowed us to make things that looked in terms of low level properties a little more like the initial image. And I guess in your reconstruction you use versatile diffusion with only the clip vectors and without this this initialization.
So actually we did use the initialization, and okay. We actually forked your repository at the time. So I mostly used these slides, simplified the slides for the sake of simplicity during the presentation, but we actually reconstruct AutoKL latents that we use to initialize a low-level reconstructed image that we then feed to versatile diffusion. However, as you can see, the retrieval metrics using AutoKL were less good in our case, and I think this stems from the fact that we're no longer doing image viewing, but movie viewing. And so even though the participants were instructed to fixate the screen, there's a lot going on from a retinotopic perspective, and I just don't think we're as good as you can be when it comes to reconstructing low-level features. And then mostly, for the most part, reporting metrics on latent representations instead of reconstructions is also something that we did by design. So one thing that I'm advocating for in some of the reviews that I've been doing for the past few weeks is to actually report these metrics on latent representations, because had we been using a different generative model and computing metrics on these reconstructions, using a different generative model wouldn't make us better or worse at decoding in general, because most of the only thing that we decode is the latent representation. But using different generative models would yield different metrics, and so I'm not against evaluating metrics on reconstructions. I just think that in general it's absolutely it's an absolute necessity to report metrics at the level of latent reconstructions.
That's fair enough. I mean, it depends. The quality of the images will depend on the generator that you use, the diffusion model that you use. On the other hand, the quality of your latent evaluation will also depend on which latent you choose. You chose clip, you could have chosen something else, and you might have gotten something very different.
Absolutely.
Okay, talking about choices, so you mentioned in your presentation that one way that you can evaluate the quality of your actually of your clip latent predictions is to caption. And I thought, I mean, sure, it's definitely one way, but it's a choice. Going from the clip to text with a generative model is exactly an orthogonal choice to the one we make when we go from the same clip latent vector to an image. And given that the subject is actually viewing images, why would the choice of captions be more relevant?
So thanks for this question. There are many things I'd like to say about this. So mostly, I think it's driven by my previous answer, in the sense that I was more interested in reconstructing good latents, and then I see reconstructions as an additional result that makes things easier to understand for us humans, but that is not a necessity. And when we look at reconstructed images like the ones I've showed you, what I've observed is that sometimes the generative models try to trick us in some way. If they want to generate an animal, for instance, they make animals that don't really make sense, or you get that it's something non-human and that it has legs and that maybe it has 7 of them and that it walks or things like that. But I thought one way of regularizing this is to go through text, and then maybe you get sentences that are ungrammatical and that, but you can't really make up words. You will end up generating sequences of tokens, and the number of tokens that you can generate is actually finite, and you'll end up in this space. So we mostly saw this as a way of advertising the fact that obviously monkeys were not seeing text, and we can generate something that's completely different. But also that maybe these reconstructions would somewhat not have some of the bad properties of image generation. But again, that really is a choice. You're right.
I want to save some time to go to a few more questions on a larger scale, or more abstract questions. And actually, since you're on this slide, this brings the question of, now that you're able to functionally align human data to a monkey brain, is there a way that you could use this to discover regions that we might not expect to see in a monkey brain, or to discover the premises of these regions? I'm thinking of things like the visual word form area or the number regions, where we may not expect monkeys to have those, but because humans have them, and because you have a plan to go from the human to the monkey, you could actually get a first idea of whether there is something, or whether all the mass has been transported to some other place and it doesn't make sense anymore.
So very frankly, this was obviously one of the hopes of such a metric, of the optimal transport problem that we presented in the first slides. I've talked about the things that worked. I think I should also talk about the things that didn't work. So one of the hope with the fugue metric was that where is it? Yes. One of the hope with the fugue metric was that the unbalancing constraint could help us identify parts of the distribution of signal in the monkey or in human participants that cannot be efficiently mapped between species. Things did not happen this way, in the sense that there are some areas that are more or less transported, but it's not interpretable in the sense that I would have liked in the first place. I think this might be an impediment of the model. Maybe this loss is not, is not the right tool to model these differences. So what we've tried is to unsmooth this constraint, and rather than using a Kullback-Leibler divergence, use L2 norms, etc. So we've tried many different setups, but we could not come up with maps that would tell us in the end, oh, okay, this is a new functional part in the human brain. I think all hope is not lost at this stage. One of the interesting properties of fugue is that it benefits from the good properties of Gromov-Wasserstein distances. And one key ingredient here is that this matrix D is the matrix of geodesic distances in the source brain, and we have a similar matrix in the target brain. But we could use other matrices that are only well defined at the scale of participants. And so one of the objects that I'd really like to try and explore is are matrices of white matter tracks connectivity between species. And I think this is I think an important ingredient to add here, rather for interspecies comparisons rather than distances on the cortex. I think, again, we lack, when comparing humans and monkeys, we lack protocols that make sense in both species. Resting state obviously is one simple endeavor, and we can try and collect a lot of data using this protocol. But again, functional connectivity matrices are only well defined at the scale of participants. And again, luckily with this sort of Gromov-Wasserstein constraints, we can easily plug these objects into a distance that makes sense across participants. So long story short, I would love to have an answer to your question, and I think it's still open. We've tried many things that didn't work. I think we tried the most simple things, but now there's room for collecting more data and plugging them in solvers that are really similar to what I've presented here.
Going in the same directions. So maybe one interpretation is that the monkey brain is at least functionally too far from the human brain, and so there are questions that don't really make sense or that the method cannot address. But what about an abnormal human brain? And I could imagine using a combination of FUG and brain cockpit as a sort of a diagnosis tool or something, I mean, for clinical diagnosis, where you could basically take an abnormal brain or a patient's brain and, try to figure out what's different in this brain compared to a human brain, and what characterizes, I mean, I don't know, different types of clinical diseases. Have you thought about this?
I'd love to do this. Unfortunately, Brain Cockpit has 2 users at the moment, so we need we need a bit of traction maybe to start these things. But obviously, it's something that I'm thinking about. I think maybe the easiest venue with the current state of this work would be to use the fugue distance as an indicator that someone is maybe pathologically far from a prototypical brain. And again, this is not clear what we can do at the moment because I've only seen participants who were a priori healthy. So I haven't really seen be given images of patients. So I don't really know if the value of fugue could be an indicator that someone is made different in a way that would require diagnosis. But this is really easy to implement, and hopefully, I think the hardest part in that would be to decide on what kind of fMRI protocol we want to do with these people to minimize acquisition time while maximizing recall of unhealthy participants. And this remains an open question. I hope that resting state can bring some answers, but I doubt it will go all the way. Hopefully that makes sense.
I don't know if I'm allowed one last question. I'm asking the president, maybe a short one. It's a very generic one, so, well, I'll go ahead. Maybe this doesn't make sense, but do you think there's anything against the idea of using FUG to align fMRI data with another modality like MEG or EEG, or, I mean, of course you need some temporal alignment and so on, but in theory, would it be possible?
So maybe an intermediate thing that we've tried in the lab is to use fugue to align EEG to EEG data. But I think it's kind of an overkill. It didn't work really well. I think you mostly just need rotations of channels, etc. I haven't tried and haven't thought of using fugue to transport distributions of fMRI signal to other modalities. And maybe as the main user of fugue, I would probably not recommend it. I would love for it to work. but I think I'd go with maybe more brutal methods where, you know, I would try and design maybe an encoder for each modality and try to make sure that both encoders end up in the same latent space, and see if I can somewhat, I don't know, inverse these encoders and see if I can derive properties either functional or anatomical from these encoders. But I think Fugue, you know, needs to be carefully tuned and can be brittle when used in different setups. And so I'm a bit skeptical that it could shrink an entire brain scanned with fMRI into a limited number of channels with MEG or EEG, for instance.
Okay, well, thanks a lot for your answers.
Thank you.
Thank you very much, Ruffin. Now I'm giving the floor to Wim van Doffel, Professor Wim van Doffel from the Catholic University of Leuven. Wim, you have around 10 to 15 minutes for your questions.
All right, thank you well, Sunil. Very nice work, Alexis. As you know, I'm very, very interested in this kind of stuff, and definitely the monkey-human competitive stuff. So we endeavored on that quite some time ago, and then we let it go. And I'm very happy to see that sophisticated methods are now being developed to do this, you know, in a sensible way, not just like in a way that a poor man would do it as we did at that time. I also think that your presentation was very, very clear, very well structured, made things more clear than when I read your thesis. But obviously I also have to ask you some questions, and some of them maybe some nasty questions. So I apologize in advance.
Go ahead.
And actually some of the questions I had were already asked as usual by Emma and Ruffin, the ones that asked questions before me. The big elephant in the room, I think, are the hyper parameters, right? How to tune it, and I think you did some effort, and you mentioned it also in your presentation that you can walk through the hyper to the different parameters. The big problem is the interdependency between the different parameters. So if you change one, you will affect the other one. So it will be very, very difficult to get to an optimum. And yeah, I want to hear from you how to deal with this issue, because definitely the spatial relaxation, basically, I think is the important one. This is sort of like the first big advancement that you made, I think, compared to sort of like the hyperalignment from Haxby. But how would you go about? Because I think there are, of course, definitely for the interspecies comparison, we know that at some places probably things are conserved, in some places things change, and some brain areas do not exist in a monkey, and maybe vice versa. So how would you go about dealing with this very heterogeneous spatial shifts in functional properties in particular parts of the brain?
Thank you, Wim, for that question. So there are many things to say, I think, at this stage. First of all, yes, hyperparameters are hard to tune. And mostly once you've found a good combination of values for each hyperparameter, it's only valid given that the number of vertices in the mesh does not change too much. So what I did is I fine-tuned all of these hyperparameters in human brains using surfaces of 10,000 points. And then what I would do is, obviously, when you compare humans and monkeys, their meshes have different numbers of vertices. I would usually sample 10,000 vertices in the monkey brain and in the human brain, so as to make sure that I fall back into a setup that I've mapped and that I know. So this is what I've done. But now if I were to redo things, and now with the information that we have, I think that actually the anatomical constraint in the way that we use it interspecies is not the way to go. I think we've tried many things, and it does bring some improvement compared to hyperalignment in some setups, etc. But I think now if I were to redo things, I would not use the geodesic distance in the cortex, but rather more richer, more informative data, like white matter tracks. And these are not perfect, obviously. It's noisy. but it's going to be a much nicer way of describing the cortex, especially across species, than what we do at the moment. And obviously, if we do this, we need to somewhat start from scratch again. We need to evaluate the relative importance of each of these contributions, because now this one has different distance matrices, etc. But that's the thing I would do. I think.
But
I would
I
also,
think sorry to interrupt, but I think the biggest problem is that it might work very well in particular parts of the brain, but not very well in other parts of the brain. I think you showed an example, and I think you showed it when Emma was asking questions. The slide 35, I think. So where 35?
was it there?
This one, I think. Yeah, this one. Maybe from an anatomical point of view, this makes sense, but from a functional point of view, this does not make any sense.
Yes, I understand.
These are points in the brain that we do know, we have the information that do not match in the human and the monkey. So this is because in the monkey, primary visual cortex is sort of like moved to the lateral part of occipital lobe. And that's exactly what you're looking at. This is sort of like 3 degrees eccentricities along the horizontal meridian in V1, which is not the case in the human brain. So we know that's a problem there. So my question is like, how would you, given some of the knowledge that we have here, how would you deal with that, or what kind of information would you need to solve this? I think basically epsilon issue to some extent.
So again, that's a very interesting question, and I think we're at the core of like the information we've gathered with this work. So I think you're one of these people that I think I'm very happy to share this information with. I mean, you're the one person I want to talk to about this. I think mostly if I were to start fresh, I would start by removing the anatomical constraint. So starting with only functional data.
Yeah.
And then from there, I would ask myself, what kind of data do I need to get richer representations, richer.
I think you're again frozen, Alexis. This is just me, Rufus? Sorry, you are frozen. Sorry.
Oh yes, sorry. I think I think we were at for a few seconds.
Yeah.
The connection stops every 30 minutes for 30 seconds.
No problem.
We're back. Okay, so mostly what I was saying is any data. I would rather go on the data collection side than on the model side. I don't think using geodesic distances on the brain is going to solve this problem. I think it was a very nice try and very intuitive, etc. But it won't.
Was that 30 minutes?
It's not me, Rufus, right?
No. It's stuck for everyone.
Yeah. I think they want to go to the reception.
Okay, we totally lost them. Oh no, they're back.
Okay. Yeah. Right. We know the image. So just go ahead, Alexis, because you were frozen again.
Sorry, I really want to answer your question, but
okay. Yep.
All right. So, can you hear me all right? Can you see my screen?
Yes.
Okay. So mostly what I was saying is, I think, I wouldn't go too much into the model side of these things to try and find an answer. I think the data that you and your lab have collected is just a tremendous amount of information that we, for instance, and I mean, we at INRIA haven't even grasped. And I think if we were to use resting state data, retinoscopy, white matter tracks, you know, without more complex models than this one, this could already bring really nice improvements, I think, to the kind of methods that we have here. Also, I think, you know, one thing that was a bit hard in this work was to convince both people who were competent in neuroanatomy and people who were competent in machine learning. And I think with the kind of data-driven approach that I've chosen, the ML people were, you know, quite convinced. But obviously, when I show these images to people like you, you're like, obviously, you notice that these two parts should not be matching in the two species. And I think to some extent, we need to be okay with the fact that if it's data-driven, I don't think we'll find a perfect mapping in the brain. But I think we can align very well some parts. And, you know, there will always be noise around. Again, I'm not making this a principle, but it's just empirically, this is what I've been seeing for the past few years.
Okay,
functional
so
alignments.
so your answer to it is like just add more data, and I think it does make sense to add, for example, rhythmotopy data, because
Yes.
not all of the brain, of course, is rhythmotopically organized, but this definitely will constrain some parts of the brain in a much better
Yes.
way than you did so far. I fully agree with that. But this brings me, because I don't have too much time, I guess, this brings me
Sorry.
to a point that you made in your conclusion. You said that it works better with just like a static image kind of paradigm where you just flash faces, or you know, like static rather than the natural movie viewing thing, which is very surprising to me. Is this because of the TR, you know, that you have too much information within a given TR, that you're sort of like screwing up the signal that you're collecting every TR in a movie, which you obviously don't have with static images? Is that the issue? And is that what you're referring to, optimizing the sequences, the MR sequences, or having other modalities would be very beneficial there? Because I cannot believe that natural movies would not work better, theoretically at least, in this kind of analysis.
So I'll try to go quickly, but again, thank you for that question. I think mostly what I'm saying is an intuition developed on what I've seen in human participants. It's easier to decode if they're watching images than movies. And I think maybe one reason is that, contrary to what happens with monkeys, human participants in these experiments might not have been fixating, you know, exactly the right way, etc. There are confounders that are a bit hard to deal with. Then also, since we use latent representations as pictures, but also movies to work with, it's probably natural that it works best with images, because all of these models that we use, all of these encoders, were trained on images. And, you know, we still have trouble embedding information from movies in the sense that we don't embed movement, or we don't embed trajectories, or like there's no time information. So I think we actually, you know, the kind of information that we're trying to regress in these approaches is just not optimal for movies. I think this is going to change, and the machine learning world might bring better models very soon for these modalities. But right now, it's I think it's doomed to be this way for at least a few months.
All right. A more general, you know, question, like you started out in your presentation with the different ways how people have been doing this, sort of like trying to register in the functional domain rather than the anatomical domain, and you mentioned hyperalignment, the different versions of hyperalignment, and other methods, diffeomorphic, non-diffeomorphic. So did you benchmark? Did you? I didn't find it. Did you benchmark? Can you say, okay, hey guys, use this. This is slightly better than the other one. Or I'm not entirely sure. I might have missed it.
So Thomas Bazet, who was in the room, and other people have tried to benchmark these techniques. Maybe one thing I'd like to say is we've tried to benchmark MSM, and Emma Robinson and her team were really helpful in trying in helping us finding the best hyperparameters to benchmark their solver. But long story short, it's a bit hard, and it's a bit hard because it's never really clear what task you want to be good at.
Or what the ground truth is, probably.
Exactly. So the baseline is anatomical co-registration, which obviously does not exist, between humans and monkeys, which is why we developed also these decoding techniques. I mean, they could serve as a baseline when trying to benchmark alignment techniques across species. And long story short, in this case, in Thomas Bazet's work, they tried to decode, so they classified signal from different participants that had been aligned or not, but again, it's not clear whether classification or regression is the best thing to do. It's not clear whether decoding or encoding through correlation maps is the best thing to do. Maybe we need to test all of them, but then the question boils down to, how do we share code? How do we aggregate these results in some way? And maybe while we're at it, we've compared our method, our Fugue method, with MSM. I don't think we, quite sincerely, I don't think we found the best hyperparameters to tune MSM. And we've tried our best for weeks, but it may be a weakly tuned baseline problem. So one venue in the team is, some people are working on a project that is called BenchOpt, where people can just contribute results and solvers to problems that are posted online. And so if you want to compete by proposing a new solver and testing it on already defined data sets, etc., you can, but I think it will have to be a multi-lab effort, and it will hardly be a single paper somewhere.
All right. Well, I have many more questions, but I get the other people will also want to ask a question. Thank you very much, Alexis.
Thank you very much, Wim. And indeed, we're moving to the next examiner, Dr. Gabriel Perrier from the École Normale Supérieure. You have around 10 minutes.
Okay, thank you very much, Mrs. President. Do you hear me well, Alexis?
Yes.
Okay, perfect. Maybe first a few personal words. I must say I'm very pleased to be here. Also, I cannot be here in person because I've witnessed your progression during this PhD. This is very impressive. I think your defense was perfect, very nice, very lot of nice illustration. But I think more than this, I want to stress the tour de force that you've done. It's, I think, the first time that I see really Gromov-Wasserstein in action on very large scale setting, and I think this is really incredible. And I cannot overstress how it's difficult to scale this algorithm because, well, these are just equations, but of course this includes, I mean, highly difficult computation, and the fact that you can make them scale, it's I think very impressive, and also the fact that it works in practice on challenging tasks. I'm very pleased to see this.
Thank you.
I'm going to ask not so many questions, but I think probably a bit more technical question. I'm definitely not an expert in anatomy and functional imaging. First of all, about Gromov-Wasserstein, one thing that you've said is that it's useful to maintain the global structure, which I would have, which is surprising and interesting, because I would have said that usually people would more like to use it to also somehow smooth the transport plan, so to include a bit the local structure. Can you comment on how much Gromov-Wasserstein operates on a local scale and on a global scale?
So then I think I'll start from the I'll start with pictures of brains at the end of the presentation. Oops. So maybe I want to show you. Oh, actually, the ones Rufin van Roelen was talking about. So this, actually, what you see here are colorization that are transported from a human subject to a monkey subject. And I think what's happening here is the plan is so smooth and there are no local changes because the Gromov-Wasserstein constraint was just maybe orders of magnitude larger than the Wasserstein constraint in the fused problem. And so what we see here is that if the contribution of the Gromov-Wasserstein constraint is too high, we just don't make or very slight local changes. We mostly keep the entirety of the anatomical organization of the brain. However, when we relax this constraint, and I want to show you this image right here. Oops. this image, what we see here, it's a bit hard to see, but I hope you can see that some of the voxels, most of the voxels right here are red, such as comparably to what's happening in the occipital lobe in the human brain. Most of them are over there are green, such as what's happening in the motor cortex of the human brain. And so at the local scale, we see that at the global scale, we see that the big parts of the brain have been preserved, and then locally there are swaps between vertices, which is one of the properties that we wanted to have in the first place. You're right that obviously this brings some smoothing, and it's actually something we'd like to fight against more than embrace. We'd like to this part of the equation to solely bring anatomically preserving features.
And I'm wondering, so maybe it goes in this direction, that here you've made a very strong choice that you are penalizing the square of the difference of the distance, which is a very peculiar choice. It's not the one that was initially used, I think, in the Memolis paper, for instance. Could you comment on this choice as opposed to, for instance, removing the square, which would give less weight to very large distance?
I think this was first of all, I haven't tested the case where we don't square these distances. I think it was our intuition from the beginning that we would like to stress long distances more than short distances. But obviously your comment makes a lot of sense, and there's no impediment to trying to modify the solver so as to try to solve this problem without squaring anatomical distances.
Okay, I think it could be nice to have a better understanding of this parameter and more generally the way you compare distance in Grove-Wasserstein. So it's not a criticism. I think you have to make choice, or otherwise there's just too many things to test. But in my opinion, I think we need to challenge a bit probably what the father have been given to us. And I mean, already using a square is nice, but I think we could go one step further. Maybe a bit more high-level question. One thing that seems to not be using is neural network. And there has been a trend in optimal transport to try to do some kind of a wedding between neural networks to compute transportation map and also contour-rich formulation. I'm wondering if you think this is possible in your setup and this would be valuable, or on contrary, you think it's a dead end because neural networks are not really useful for this type of work and also more generally for optimal transport?
So thanks for that question. There are many things I'd like to answer about this. I think I don't want to quote Rémi Flammary on this, but I think it was also a choice from the beginning that we would work with machine learning oriented solvers rather than deep learning to try and solve these problems. But I remember you mentioning a few years ago, I think a year and a half ago, that we could do some warm start with working with deep learning models that try to approximate optimal transport plans. And I think that would be one of the reasons why I would try and use deep learning models instead of the solvers that we have now. I still have this belief that Sinkhorn algorithms and friends will be faster for the problem. Will just run faster for the problem that we have at the moment, but I'm happy to be proven wrong. And so yeah, that's it. I think with the solvers that we have at the moment, the fact that we can't really warm start is maybe one of the main reasons why I would try and pursue the deep learning approach.
Okay, thank you. And maybe my last question, so I will select one question in my list. Oh yeah, so one thing that I'm very interested in is more like going to dynamical questions, so things where you just don't want to match two distribution, but that you would like to track some evolution. And to me, the type of things that you do, it could raise also maybe this type of question where there would be some dynamics in the brain. In which case, I'm wondering whether the type of tool you have developed, so Wasserstein, of course, but Como Wasserstein is much more interesting in my view. would be useful to model in time the evolution, where instead of just saying that you want two things to be closed, you want things to minimize some notion of energy. So a bit like particle systems that would minimize an energy, but this time this would be the evolution on the brain, or maybe the brain themselves, that would be modified. I mean, I don't know if you know Wasserstein radon flow, but this would be in some sense this idea.
Mm-hmm. Yeah, thanks for the reference. I hadn't heard about that work, but I think eventually, yes, this is a very interesting venue, and this is one of the reasons why I kind of want to increase the temporal sampling in fMRI images, even though it leads to worse signal-to-noise ratio. I think using these sort of metrics on time series with more samples would be obviously a very interesting venue. And then, but for the specifics of what you mentioned, I haven't heard of these models.
Okay, so we could discuss at some point. I think there's a lot of interesting use of optimal transport beyond just registration, but to rather track in time some evolution. Okay, so I'm going to stop here, and I just wanted to congratulate you once again for the manuscript and also for the defense.
Thank you.
Thank you very much, Dr. Perry. Sorry. And now I'm giving the word to Claudia Lunghi from the École Normale Supérieure for 10 minutes.
Hi, Alexis. I start to congratulate you as well about the quality of your manuscript, of your presentation, of your work in general. I think you did really an amazing job. Everything is very clear, and the amount of work you've done, it's impressive. So I got a question actually on a study that you present in chapter 5. So in the interspecies comparisons, I'm sorry, it's not what you presented in your defense. About the different contrasts. So here you were dealing with a kind of a rich data set.
Yes.
And well, I'll focus on a visual stimuli because it's a bit more my expertise. And so you had a visual stimuli of different kinds of visual stimuli with a lot of different contrasts that you could have considered, and that you show also in the table 5.1.
Maybe here.
And then there's table exactly. So you have faces, non-faces, bodies without faces, you got objects, animals, and so on and so forth. And then actually what you show, so I was expecting like more a finer grain of a mapping that you could do for visual areas. And instead you only show the contrast between visual versus control. So I was wondering whether you've looked into the other contrasts, if you've been able to map different visual areas across species, which would be very interesting. And I'm thinking, of course, two example, very famous example, like, for example, the fusiform face area or the extrastriate body area that have been found in both species, and whether you could further validate your method by mapping these areas across species.
So thank you for that very interesting question. And so many things to say here again. Initially, what we started to do with this work was we had a limited number of contrast maps, but quite high. So we started working with them. I didn't use the anatomical constraint at first. So I tried to find again, face patches in the temporal lobe of monkeys, and that they would be aligned with the fusiform face area, etc. And this works. So you can isolate these patches. But apart from this, like the rest of the brain would be completely messed up, like it wouldn't look well. So we tried to fuse in the anotopical constraint so that we would get all these cortical areas the right way. But the conclusion that rose from there is that actually it seems like a high number of contrast maps, but it truly is not. And this is mostly why we switched to using movie watching and time series rather than this sort of setup, because we came to the conclusion that having, maybe it could be impressive to have 15 contrast maps, but actually this is not enough to distinguish properly between all areas of between all areas of the brain. So this work right here was really preliminary. We wanted to test the computer sciencey parts of the algorithm and how it would work in low data regimes in the functional space. But mostly the conclusion from this work is unfortunately not related to neuroscience, but more to computer science. It's at this stage that we concluded that we needed richer stimuli and not combinations of contrast maps. Sorry, I realized I repeated myself. Does that answer your question?
Yeah, it does. I'm a bit confused because for other, we just said just a little bit before that static images will work better than movies.
Yes, images, absolutely. So this is absolutely true, but the amount of data, there is also a bit of a barrier you need to pass. So showing images is definitely going to work, I think. Computing contrast maps from images, from image viewing, I think is not the way to go. I would instead show images to monkey participants and keep the raw or like mildly preprocessed time series collected while seeing these images instead of computing statistical maps like contrast maps. And in this particular example, we've grouped together categories of contrast maps like faces or bodies. You have examples here. So, images that are images of different animals would fall within the same category, which I think does not help us leverage all the data that we have at hand. Actually, and this is an intuition that I think we've built more and more strongly in this team. There is a lot of signal in bold signal or Mayan signal, maybe much more than we exploit when we compute contrast maps by fusing together images that we think belong to the same category.
Okay, thank you.
Thank you.
I had a couple of more general questions. A couple of them have been already asked by Rufin, so that's good for time here. We can catch up there. I'm left with one general question. So about a topic that you introduced in the beginning of your manuscript. So in the introduction, you explained very well the advantages of having functional alignment rather than anatomical alignment, and especially the importance of the inter-individual variabilities. And the importance of taking care and considering these inter-individual variabilities, for example, in a case of studies that are interested in investigating functional changes in the brain occurring with experimental manipulation inducing brain plasticity. Well, you would expect that question from me. And so I wonder, in that case, would your fugue system be flexible enough to detect functional changes, not only, to align to have a functional alignment between individuals, but also to detect functional changes across individuals, within individuals and across individuals when aligning them. And on a related question, because you conclude your manuscript kind of advocating the idea with that decoding will be the future of MRI, and it's superior to encoding models or paradigms. How, in such kind of paradigms, interested in functional changes related to brain plasticity, how do you think that decoding will be better than encoding to detect those changes?
So these are two big questions. Regarding the first one, sorry, I think. Sorry, I lost track. I lost track of the first one. Can you just repeat one or two words?
Well, in few words, it's like, would your be flexible enough to account also for changes, functional changes to brain plasticity, and what would be the advantage of using it?
So one hope when we built that quantity is that it would be informative as to how similar two brains are. Two brains of monkeys, two brains of people, but also two scans of the same person at different period in time. And so with regard to plasticity, I think, our hope was that maybe we could track, brain scans of a person before surgery and after surgery should be distant in a way that's very informative if using this metric. I haven't been able to show at this stage that this distance, that this quantity can be used as a metric. But I think this is truly an object that we should try to seek. So maybe let's forget optimal transport. Let's forget about fugue. Let's think about brain images. If we had some way to encode them, that is to say, to represent them with vectors that make sense across participants, across individuals, etc., distances in this common representational space would be of crucial importance, and they would probably carry the properties that that your question somewhat mentions. This, I think, would be the best tool we could hope for, some sort of geometry of the brain, a latent geometry of the brain. It was the hope with fugue that we could get at least a grasp of this useful distance, but quite frankly, at the moment, I don't think we've. I think we've only scratched the surface of this game. Mostly, I'll end up with two things on this question. So we've tried to see if fugue was predictive, if the fugue distance was predictive of, like, let's say we train a decoder on you and on me, or not, just on you, and the fugue distance between us is very small. We would expect this decoder to transfer very well from you to me. That's the hope. We haven't been able to show that this property exists with fugue. Hopefully someone else can do it. And sorry, can you say one word about the second question?
The second question, you advocate at the end the superiority of decoding over encoding.
So maybe the key intuition as to why I say this is when we play the game of encoding. So when we predict what is happening, when we train models to predict what's happening in the brain, we're relying on noisy samples of the brain. If we play things the other way around, we know exactly what the person was seeing. So we're trying to train a model that goes from noisy brain data to perfect representations of whatever image the person was seeing, etc. Obviously, we're missing some information. Maybe the person is ill at ease in the scanner, or etc. We're missing something that's going on in their brain, maybe in terms of perception. But I think it's going to be a lot easier to predict non-noisy representations than it is to rebuild blurry images of either fMRI data or EEG, MEG, electrodes in your case. So I think just framing the problem the other way around gets rid of a lot of the things we're fighting against when we work with encoding models, especially this thing right here, like the fact that we try and explain signal in the brain just as a response to stimuli and ditch all remaining signal as noise, I think is a major impediment to understanding what's going on.
Okay, thank you, and congratulations again.
Thank you, Dr. Lungi. My turn. A lot of questions have been already asked. I would like first to congratulate you on your manuscript, which was extremely clear, very pedagogical, and your presentation, which just confirmed what was already in the manuscript. So congratulations for all this work. I'm left with very general questions, and one of them probably pursues on what Dr. Lungi just raised, which is about the relationship between encoding and decoding. But let me get back to the start of my thought, which is the fact that the discussion and your answer right now to the previous question, enhancing the fact that you can match very easily a controlled input to whatever the brain does, thus favoring a decoding approach, is really due to the type of tasks and paradigms that you've been using, which are mostly sensory. And then when we shift to cognition, more complex cognitive tasks, which is one of the aspects that we want to understand across individuals and across species, then things becomes more blurry because obviously the intrinsic state of the brain, both from an anatomical perspective and a functional perspective, is different. So what is your thought in these lines?
This is absolutely true, and I think it's so again, I see this from a very I think machine learning-y perspective, but unfortunately at the moment we don't have good representations for the kind of cognitive processes that you are studying, and I think a conjunction of things happen at the same time. It just so is that the occipital lobe is organized in a way that we have a fusiform face area, etc. The organization of this part of the cortex makes it easy for us to study the brain. And at the same time, we've seen the advent of these deep learning models that give us very nice vectors for representing images. I don't think it's impossible to study cognition. Obviously not, but we don't have these the tools that make it easy to naturally represent what's going on in the brain and ask questions in the form of latent representations. I think my hope is that if we want to probe attention in the brain or planning, or if we want to study how one evolves through plasticity, etc., these are going to require very much richer embeddings than the ones we have at the moment. And I don't know who's going to win first. I think machine learning people somewhat expect the neuroscience people to come up with a good representation. We somewhat need a good representation from maybe from deep learning models that would solve these tasks in a way that does much better than what we have now. But yeah, so long story short, I think these questions remained widely open. They're harder to study because the brain is more fuzzily organized in areas that are involved in these processes. And on top of this, we don't have good representation at the moment. I hope that once we have good vectors to ask questions to, this shall be easier.
And then I have another question, and I will. Do you think that your model could exploit it independently from functional data?
Yes. So I think I'm allowed to do this at this stage. Oops. I would take this part of the equation right here, the green one, and actually add many green elements to the equation. One of them would try and preserve the general anatomy of the brain. One of them would try and preserve the geometry of responses to resting state data. One of them would try and preserve connections in the white matter, etc. And definitely, this is what this part of the equation was thought for in the first place, because it makes it possible to plug in resting state functional correlation matrices, if you may. Yeah.
I think that would really bring your how do you call it, FUG method to a clinically the potential of predicting clinical situations, because then you would have a general description of what can happen in the brain. So thank you very much. And now I give the word to your PhD directors, and I will start with Dr. Bertrand.
Thank you.
Thank you. We currently have discussions at INRIA about how to run good results projects and risky results projects. And then some people say, oh, we should not hire students on risky research projects because that's somehow dangerous for them if we don't achieve the goal. And then I think of you and I say, but no, that's exactly the opposite. We need new people to implement new ideas and go in new directions. And I think that your PhD thesis is exactly the illustration of that we need new people to create a new way to do research and to move forward. And I think that you took risks, we took risks with you, and we were right to do so. You have done stuff that we would not have. I would not have thought we would achieve that. What you presented today 4 years ago. Doing optimal transport with interspecies mapping, validation based on naturalistic stimuli, decoding, etc. Each of these words is a challenge, essentially. So putting everything together is really impressive. Even if, in some aspect, we just scrapped the surface, indeed. And so, obviously, I think everybody can feel that you've really done great exploration job. And because you have really peculiar skills in first in computer science, in implementing applications, designing interfaces, but also in understanding the math, going all the way from equations to code. And so doing this by collaborating with experts in optimal transport, doing the human and monkey fMRI data analysis, which is also another kind of challenge. Sustaining, developing active collaborations with neuroscientific teams and also software development teams like Nylon team, and even beyond Nylon with NearView, etc. Okay. And communicating, because research is also that. At the end of the day, we need to communicate to provide a clear picture of what we did. And obviously, you're really great at doing that. So it was a pleasure to work with you. First, because you brought the energy. In many PhD theses, you have to bring, as a supervisor, the energy. And what was great with you is that you would always come with something, with some energy and new ideas. I guess it's fair to say that you proposed the best ideas that were done in this PhD thesis. And you did something that very few students would do, which is really to bridge with other labs. You put a lot of energy in what you do, but you're also reliable. We can count on you, rely on you when it comes to showing deliverables to some funding agencies, etc. And that was not easy. And you did it mostly. We did it together, but you did it for most of the part. And finally, for the ambience you put in the team, especially during the retreats. But I think that everybody in the team really appreciated interacting with you. Of course, we have not solved all problems. We have not solved some of the challenges that we hoped we would solve, but we have solved other challenges. And I think this is really great. You have opened new ways. At least 2 PhD theses will follow in our lab. So it's just the beginning of something new, of many things. And I really would like to thank you for that.
Merci beaucoup, Bertrand.
Okay, so I guess I have the last word, and it's a great word of pleasure, I should say, to congratulate you for this, first of all, beautiful presentation, very clear document, and especially 4 years of work together to try to develop these tools and to apply them. It's exactly as Bertrand said, I think this work went way beyond what Bertrand and I expected. In many ways, I think I started it by showing you the Montini and Van Duffel paper, which, by the way, Wim is here. I really admire this paper. It's very inspiring. But, for 10 years, I was thinking there should be a mathematical method to do it at the whole brain level. And so I showed you this problem. And here you are 4 years later with a very elegant solution. I think you brought indeed many of the solutions. You brought the optimal transport. You were inspired, of course, by Bertrand and the Bazet paper. You also did an internship of a few months at the fair, and you came back with decoding, beautiful decoding solution with Jean-Remy King behind. So that were things that I did not expect from this PhD, and it was really very exciting. I have also to mention, if there are any potential employers in the room, the incredible speed and quality of the programming and the mathematics that you are applying, including the development of apps that actually work and have a nice interface. It's just amazing. So the brain cockpit, I think, is a really amazing achievement just by itself, an engineering achievement. Exactly as the others have said, I think this is opening many doors, and we still haven't looked what's behind the doors. So there is a lot more work to do. There are several questions. One is should we align in cortical space as MSM does? Should we do high dimensional alignment like in hyper alignment? Could we just do one voxel to another voxel alignment? Apparently Nancy Canwicher, by the way, Nancy just got the Kavli Prize, which is a super important prize together with Doris Sau and English Firewall. So we're very happy for them. But apparently just mapping one voxel onto another voxel may already be sufficient to get some of these correspondences off the ground. So maybe that's something we should have had as a baseline. Yeah. So my little bit of frustration is that many of my original questions in neuroscience are left unaddressed or not fully addressed, at least. Let me ask you one of them. Do you think that the same matching parameters should be applied for the whole brain as we are doing now? One of the things I wanted to ask is whether some areas like frontal cortex maybe have a very different principle of mapping and are much more intermixed compared to others. You have evidence for that?
Evidence, no, but I have intuition. And I think the question was raised by Gabriel Perez from the first comité de suivi individuel. So the thing we do with optimal transport at the moment is we just give every voxel a chance. You know, like we give every of them a chance and an equal chance. I think obviously we have priors as to what part of the occipital cortex of the cortex are already exist in the monkey brain and do not. And so tweaking the values for these little omegas here would help implement these priors. But again, I think maybe this is not the most interesting venue as to what we want to do. I think maybe what's behind your question is I think if we want to implement different strategies for different parts of the brain, maybe what we want to have in the end is some way to merge transport plans together, compute some of them with some constraints, compute others with other constraints. And I don't think we want one big optimization problem that tries to solve everything at the same time. We can make different hypotheses when we use retinoscopy data, when we use white matter tracts, etc. Maybe what we need is a way to define a mathematical object, which would be in some way a fusion of these transport plans in a way that makes sense. But I don't think I can come up with an idea at the moment.
Maybe we'll be there in 20 years, but I completely agree. For instance, the retinotopic maps have already been aligned very nicely. Maybe not fully, but very nicely, the work of Kolster, Wim, Chi, and others. So we should somehow have found a way to take that into account and align the other parts that are not so well known. So that would be great to achieve such a tool. Yeah. Some of the questions, I just want to list them also for my own memory, because we started with so many good questions, I think. One question was whether there is really a diffeomorphism from one brain to another, even from human brain to human brain, or whether there are some discontinuities in some places that suggest that the areas are really not the same. Maybe there are some missing areas, surely from monkey to human or human to monkey. There's going to be some missing areas. Do we actually map the whole brain? So I really regret that we haven't looked more into the transport plans one by one. And you provided a tool for that so that we could see which areas are being transported and which are not, or which are being intermixed, perhaps even against what we already know in the literature and which are not, and so on and so forth. Yeah. So my last question was, how do you leave this tool? Do you leave it in such a state that a student, maybe in neuroscience now, could arrive and take it and ask some of these questions? I think you do, right?
I think the tests are passing. Let me check.
Do this in real time.
But yes,
I did not prepare this.
All of these tools have been published online. They're tested, and there's hopefully a release that's in a stable version. So in this particular case, you know, a student coming in could just pip install Fugue, and they would get a
tool. Yes, but what about the maps, the optimal mappings that you found? They are in brain cockpit?
No, they're not. So they're not, because the version we have at the moment that's online and that's featured by the eBrains project only has IBC and was made for this purpose. Someone coming in would have to recompute these maps and put them in, probably in a different instance than the one we're using here.
Okay, so maybe that's a little request that maybe we should do that as we are also publishing this work. Maybe we should publish it with some kind of tool that would allow others to ask about the quality of these mappings and whether they make sense for every area and so on. Anyway, that's my only regret, and it's not a regret because it shows there's so much more that we could do from what you have done. So I think this is really a very, very interesting and exciting PhD thesis. Thank you.
Thank you.
So this, thank you very much, Alexis. This brings the defense to a closure. So maybe we can thank Alexis again, and then the jury will isolate, make a decision, and come back to you with the outcome of our discussion.
I think we ask people to leave the room.
Yes.
On repasse on a dit ça va kill le truc. Donc, on va le faire depuis ton ordi.
Ouais.
Moi, je vais bien monitor et on va pas me transférer moi.
Donc, je fais
Donc, fais breakout room, create room.
manually.
Ouais, vas-y. Yes. Ok. Tu peux, ouais, la créer, vas-y, open.
Tu veux faire? Vas-y.
Non, non, c'est. Tac. Ok. Assign to one. Assign to one.
So we're assigning everyone to a breakout room so that the deliberation will not be accessible live on YouTube.
Join room. C'était la récré. Donc normalement, c'est bon. Je vérifie que j'entends.
Ouais, vous voyez, merci. I just want to thank you again. I want to apologize. I will have to leave rather quickly after that. But thanks very much for all the work. And I hope we keep in touch and maybe apply this tool, compare it with other things. You know, it'd be great. So, Wim, I hope to see you another time.
Yeah, sure. I'm sorry that I couldn't make it
No, of
today.
course.
Yeah.
But another time, that'd be great. Yeah.
Yeah, sure.
How are you? Ils vont pouvoir nous voir ou pas? Voilà, on se met tous ici, hein, c'est peut-être mieux. Là. On va se mettre devant si on se met devant la caméra. Voilà, venez par ici pour qu'ils nous voient sur la caméra. On va pousser les micros.
Allez.
Yann, si tu viens par ici, ouais, voilà, comme ça ils nous verront. Tout le monde nous voit. Et le candidat, ouais, c'est peut-être le plus important. Alors, rapproche-toi encore un tout petit peu. Tu vas être sur l'écran. Voilà, encore un petit peu. Bertrand, viens là. Et Juliane au milieu.
Faut que je retrouve le serment.
Oui, alors tu dois lire. Je peux l'appeler par cœur, mais oui.
Avec la main sur le cœur. Mais justement, on disait tout à l'heure qu'il faudrait faire au début de la thèse le serment. Sur l'intégrité scientifique.
C'est une suite. Quelque chose. Bien.
OK.
Si tu parles là-dedans aussi, ça marche.
Ça suffit. So thank you all to be back in this room. The jury has met, discussed the presentation of Alexis Thual in front of us here today. And what the jury would like to express is the fact that we unanimously found that the presentation was extremely clear, even though it touched on very complex topics. So this is clearly a very strong skill that is useful in academia and beyond. So congratulations on this. We also noticed and want to highlight the breadth of the skills that you developed during your PhD and that you demonstrated in front of us today, from mathematical skills to coding skills to computational skills to neuroscience skills. So that's quite a lot. We also noted the very strong and genuine enthusiasm that you demonstrated during the answer to the questions, which is exactly what the spirit of science is about. So congratulations on this. And we also would like to highlight something which is important in academia and science, which is the fact that you showed a very strong understanding of the limitations of your work, the limitations of the field. And this is exactly what one needs to move forward in the acquisition of knowledge. So again, congratulations on this. And as a summary, the jury would like to congratulate you by giving you the degree of doctor in neuroscience and applied mathematics from the Université Sorbonne. There are no grades delivered anymore, but we would like unanimously to congratulate you and present you with the félicitations du jury. We've discussed among us, and we think that your PhD is clearly among the top 5% of the PhD that we get to evaluate. So again, congratulations on this. So now, thank you. So now I would like you to ask you to read the oath of the doctors in science.
En présence de mes pairs, parvenus à l'issue de mon doctorat en neurosciences et ayant ainsi pratiqué dans ma quête du savoir l'exercice d'une recherche scientifique exigeante, en cultivant la rigueur intellectuelle, la réflexivité éthique et dans le respect des principes de l'intégrité scientifique, je m'engage, pour ce qui dépendra de moi, dans la suite de ma carrière professionnelle, quel qu'en soit le secteur ou le domaine d'activité, à maintenir une conduite intègre dans mon rapport au savoir, mes méthodes et mes résultats.
Congratulations.
If you want to say a word.
Tu veux dire un petit mot?
Oui, j'ai plein de mots à dire. So I'd like to start with the members of the jury. Claudia, I think you're the person I've interacted.
Bad luck. We're at the 30 minutes.
Emails, for responding to all of our emails and sharing this data with you. I truly think this is the workhorse of the kind of research that we do. And I hope to continue our collaboration in the future. Thank you, Gabrielle, as well, for being part of my comité de suivi de thèse individuelle and for all the rich ideas that you've contributed to this work every time I needed to. Thank you, Suliane, for being the president in this work. And again, we're not closely linked, but I've met you through Mathilde, whom I have met in Wim's lab, and this is how I got to read your work, etc. And so I'm really lucky to have collaborated in some way with all of you. Et après, je vais passer en français. Stan, Bertrand, merci beaucoup, vraiment du fond du cœur, de m'avoir accueilli dans vos équipes. Vous m'avez toujours donné beaucoup de liberté. J'ai eu la chance immense de mener une thèse privilégiée où il m'a été donné de voyager comme je le voulais dans d'autres laboratoires, de pouvoir aller passer du temps sans but particulier autre que rencontrer les gens avec qui j'ai interagi. Et cette liberté ne s'est pas faite au détriment de l'encadrement. Vous avez été présent longtemps et souvent. On s'est vu toutes les semaines. Vous avez lu mon code, vous m'avez guidé dans les papiers à lire, vous avez encouragé et contredit des intuitions. Je pense que c'est le summum de ce qu'on peut attendre en termes d'encadrement. Je voudrais raconter 2 anecdotes en passant. J'avais pas prévu de faire une thèse, j'ai contacté Stanislas sur Twitter et c'est comme ça que ça a commencé. Et la deuxième anecdote, c'est qu'au 2e jour de ma thèse, j'ai présenté des résultats absolument parfaits à Bertrand parce que j'avais testé mon algorithme sur le training set. Je comprends que Bertrand ait eu certains doutes quant à ce qu'on était capable d'accomplir ensemble sur ces 3-4 années. Merci de n'avoir pas douté. J'aimerais évidemment remercier toutes les personnes avec qui j'ai collaboré, mais pas seulement à l'étranger, mais je veux dire ce qui m'a plu dans cette thèse, c'est celles et ceux que j'ai vus au quotidien. Je pense d'abord à Thomas Bazeille, je ne sais pas si je le vois ici, avec qui j'ai passé de nombreux après-midi à regarder les données au début de ma thèse. Avec Rui, franchement, Rui, les meilleurs moments de ma thèse, c'était les journées qu'on a passées ensemble au Collège de France à débugger le solver ensemble. Il y a Thomas Diguero peut-être dans la salle aussi, avec qui j'ai passé de nombreux après-midi. Merci pour ça. Pierre-Louis plus récemment. Et c'était sincèrement le sucre et le sel de mes journées. Et si jamais je fais de la recherche, ma vie ou ma vie de la recherche, je chercherai surtout des copains et peut-être des résultats. Et je voudrais évidemment remercier ma famille à qui je dédie ce travail. J'aurais beaucoup aimé que mes grands-parents puissent le voir, mais je le dédie à ma mère, mon père et mon frère. Et voilà, je suis très content de pouvoir vous montrer ça aujourd'hui. J'espère que les explications étaient, au moins sur le papier, étaient claires. Et merci, Pauline, pour ton accompagnement, toutes les discussions qu'on a eues ensemble ces dernières semaines. Ça m'a beaucoup aidé et je pense que ça a contribué en grande partie à la qualité du travail que j'ai présenté aujourd'hui. Merci à vous toutes et tous qui vous êtes déplacés et qui avez suivi cette thèse avant d'avoir du champagne. Je pense qu'on devrait le mettre avant, mais enfin, il vous attend dans la pièce d'à côté maintenant. Merci à tous et à tous pour votre temps. C'était vraiment un plaisir.
Bravo.
Jean,
Congratulations, Alexis.
merci beaucoup.
Bravo, c'est vraiment formidable. Merci.
Félicitations, Alexis. À bientôt.